Perfect. Okay. Well, welcome everybody to our February Dunleavy lecture. Just a few center updates. The Spring 2026 Seed Grant reminder applications are open for the Dunleavy MFM Center Spring 2026 Seed Grant Program. To remind folks, the center will fund two awards, $35,000 each for 12 months starting July 2026, with an application deadline Monday, April the 13th at precisely 11:59 p.m. Full details on the website. Uh, for just a lecture that's coming up under our PRISM grant, we were fortunate to have Dr. Corey Abramson, who's an associate professor of sociology at Rice University, will be speaking on his work as the co-director of the Center for Computational Insights on Inequality and Society. It promises to be a really interesting lecture related to PRISM, and we hope everyone takes the opportunity to sign in and listen. Our next lecture will be on March the 12th, and we'll be hosting Dr. Yalda Afshar, who's an associate professor of obstetrics and gynecology at UCLA, and she'll talk about placenta accreta spectrum, a sticky situation when womb mates overstay their welcome. It's my honor today to hand over the introduction to Dr. Mana Parast to Dr. Virginia Nguyen.
Welcome, everyone. Thank you for attending our Dunleavy seminar series. And it is my distinct pleasure to introduce Dr. Mana Parast, who is, I consider, one of the world's premier trophoblast biologists and a go-to person for anything placental pathology. Mana received her undergrad degree at the University of Rochester. And this is where it's always fun to read people's CVs and learn things about them that you hadn't known before. So Mana, you and I overlapped at University of Rochester and didn't even know it.
That's amazing.
Yeah, I did my MD PhD at University of Rochester. Mana from University of Rochester did her MD PhD at University of Virginia, followed by her pathology residency at Emory and a clinical fellowship in women's and perinatal pathology at Brigham and Women's Hospital. She has been at UCSD and raised through the ranks to full professor, and has also developed a perinatal research center and is co-director of that at UCSD, and has gathered a tremendous group of investigators in that arena. Her scientific contributions have been awarded and recognized by the Society of Reproductive Investigation, where she's received the President's Achievement Award, as well as her recognition by the International Federation of Placental Associations, having received the award in placentology. With that, I will turn it over to Mana to tell us about her thoughts and perspectives on placental research.
Thank you so much, Virginia. Thank you, everyone, for being here and for the invitation to speak to you today. So let's see if I can advance. There we go. And you guys can see this okay, hopefully. So just a quick overview of what I'm going to talk about today. I'm going to give you a quick overview of early placental development and implantation, which I'm sure you don't need. Then I'll jump right into how we're using pluripotent stem cells to model the human implantation site. And then at the end, I'll touch on how, more and more as, especially as I advance in my career, I'm trying to sort of integrate different aspects of my life, including my clinical work, into my research. So early human placental development and implantation. Again, just a quick overview that I'm sure you guys are all, more expert in than me. So as you know, trophoblast lineage is the first lineage to be specified in the embryo. So when the blastocyst when cavitation happens in the morulus stage, you get a blastocyst stage embryo, which has on the outside a trophectoderm, and on the inside an inner cell mass. The inner cell mass will give rise to the embryo proper, as well as the extraembryonic mesoderm elements of the placenta, while the trophectoderm layer gives rise to all the trophoblasts or epithelial portions of the placenta. Very crudely, the differentiation of trophectoderm takes place by initial expansion into a cytotrophoblast shell, where these cells are sort of deemed the stem cells of the placenta, and they are bipotential, being able to differentiate into both an extravillous trophoblast or EVT lineage, where these cells express the non-classical HLA-G molecule, secrete MMPs, and remodel the implantation site. They are highly invasive cells, kind of akin to cancer cells, and can remodel maternal vessels. Whereas within the floating villus structures, these cells differentiate into syncytial trophoblasts, which are multinucleated. They secrete HCG and are involved in nutrient and gas exchange and pathogen evasion, among other tasks. So at the implantation site, other than trophoblasts, there are a lot of other cell types with which the extravillous trophoblast in particular interacts with. These include the endometrial stromal cells and a lot of maternal immune cells. In particular, the decidual NK cells are a cell type that are enriched in this immune environment and help direct the extravillous trophoblast in their invasive capacity and in the remodeling of the maternal spiral arteries. And this is sort of, the main job of the extravillous trophoblast in early implantation and placentation is remodeling of these maternal vessels, which prior to remodeling, they have this smooth muscle layer. They have sort of they're known as being low capacitance, high resistance vessels. But following remodeling, by which the extravillous trophoblast actually induce apoptosis of the vascular smooth muscle cells and remodel and replace the maternal endothelial cells in these vessels end up becoming sort of high capacitance, low resistance vessels that can now carry a lot of blood to the growing fetal placental unit. When this process doesn't happen correctly, the lesion that as pathologists we see under the microscope in the maternal vessels of the placenta is a lesion that's referred to as decidual arteriopathy, or what is shown here is a severe form of it called acute atherosis. So you see fibrinoid necrosis in the wall of the vessel, as well as macrophages that contain lipids. And as a result of this abnormal spiral artery remodeling happening early in the pregnancy due to abnormal EVT function, the pattern of placental injury that you might see in a placenta at delivery is called maternal vascular malperfusion. And this is characterized by areas of infarction, accelerated villus maturation. And this is characteristic on the clinical side of what you might see with correlating to maternal hypertensive disorders of pregnancy, such as preeclampsia, plus or minus intrauterine growth restriction. So my goal ever since I established my lab here at UCSD has been to be able to model these abnormalities that we see in the human placenta. As you all know, we don't really have good animal models that recapitulate the human placental architecture and individual cellular function. And as such, we have turned to pluripotent stem cells. I'm going to start with kind of a quick run through of the work that my group has done, and now other faculty that we have been able to retain here at the Center for Perinatal Discovery at UCSD, working on iPS-derived trophoblast stem cells and using them to model disease. So when I first arrived at UCSD, there had already been a lot of publications showing by various groups showing that pluripotent stem cells can, in addition to giving rise to embryonic stem cells, they can also, or sorry, embryonic cell types. Human pluripotent stem cells in particular seem to be able to also give rise to extraembryonic lineages, including the trophoblast. And in particular, multiple labs had shown that treatment of these cells with BMP4 would lead to formation of multinucleated cells in culture that secrete HCG, and a subset of them would also express HLA-G characteristic of extravillous trophoblast. So early on, my first CERM-funded project, we were able to show that this process actually happened in a p63-dependent manner. P63 is a marker of the cytotrophoblast stem cells within the placenta, and we showed that by knocking down p63, BMP4 treatment did not lead to formation of these mature trophoblasts. Subsequently, along with Dr. Hori, we established a two-step protocol for differentiation of pluripotent stem cells, first into a relatively uniform cytotrophoblast-like state. And this was done by the addition of IWP2, which is a Wnt inhibitor, which inhibits mesoderm induction, which also happens downstream of BMP4. And then subsequently, we couldn't keep these cells going at the beginning, but we could convert these cells, further differentiate these cells into syncytial trophoblast and extravillous trophoblast by continuing BMP4 treatment in the presence of feeder conditioned media. And this was advantageous because it was consistent and was a consistent and uniform way of differentiating pluripotent stem cells into trophoblast, and it separated. It allowed for a separate study of trophoblast lineage specification in step 1 and terminal trophoblast differentiation in step 2. So this is just showing some of the data. This is after the first step, you get a uniform p63, CDX2, EGFR, TEAD4 positive cytotrophoblast-like population. And subsequently in step 2, you would get differentiation into two distinct cell types that were mixed in culture. The syncytialized, the multinucleated HCG producing syncytial trophoblast, and the HLA-G expressing extravillous trophoblast. And then once the Okai paper was published showing that we could derive trophoblast stem cells from early gestation placenta, we wanted to see if we could apply that media to are cytotrophoblasts that were derived from these pluripotent stem cells and derived trophoblast stem-like cells. Just as a caveat here, we actually used a little bit of a modified Okai media, which was developed in the laboratory of Jack Kessler at Northwestern, called ICTB media. This was they reprogrammed terms cytotrophoblast into trophoblast stem cells using various factors, and they applied this media, which is just kind of like an expanded version of the Okai media. And so we applied this media to our pluripotent stem cell-derived cytotrophoblast, and we basically, after about 5 passages, could get cell colonies that were similar to the primary Okai trophoblast stem cells, which they had shown could be derived from both early first trimester trophoblast, early first trimester placenta, and blastocyst stage embryos. We took these cells, we compared them, their transcriptome by bulk RNA-seq to primary trophoblast stem cells, including 2 lines that we had derived here from 6 to 8 week gestation placentas. And we compared them also to the pluripotent stem cells and the ones that were being treated with BMP4 IWP2 in step 1 of our differentiation protocol. And what we basically saw was that these cells, at least transcriptomically, were very similar to primary trophoblast stem cells. And we could differentiate these cells by applying the Okai protocols for differentiation into both syncytial trophoblast and extravillous trophoblast. And we showed, again, based on expression of various markers, HCG expression and HLA-G expression in EVT, that these cells, the differentiation was very similar to what we saw with primary cells. We also injected these cells, just as Okai and al. Had injected primary TSCs into immunocompromised animals as to see if they form trophoblastic tumors. And we, in fact, saw formation of similar trophoblastic tumors with a necrotic center with our pluripotent stem cell-derived TSC. So at the same time as we were doing this, the sort of stem cell field was moving rapidly forward with the development of, with the conversion of cells from a primed state of pluripotency, which is sort of the standard way that stem cells are cultured, to what was called a naive state. And the idea in general that BMP4 could, in fact, convert a primed state of pluripotency to trophoblast was a bit antithetical, especially in the mouse stem cell field, because it's known that definitely in mouse, by the time the blastocyst is formed and the trophoblast lineage is specified, the conversion of ES cells to TS cells just does not happen outside introduction of, outside manipulating gene expression, either by CDX2 overexpression or OCT4 downregulation. And just conceptually as well, it doesn't make sense for a cell that is in the primed lineage, kind of past the stage of trophoblast specification, to be able to generate trophoblasts. So this concept of the generation of such cells was challenged, even though multiple people had shown that, including our lab, had shown that this could be done. And just to give you an overview of sort of what happens in, or what is thought to happen in context of naive conversion of primed stem cells. Basically, this is showing using one media, but there's multiple medias that have now been developed for naive conversion. And the idea is that basically what you're doing through this conversion is that you're kind of sending the cells back further in time towards a totipotent state, where basically one of the major features of the change that's happening in these stem cells is loss of DNA methylation, including the cell type specific imprints. And so 2 papers that were published sort of back to back in 2021 in Cell Stem Cell basically showed data to prove that only naive cells could be converted into bona fide trophoblast stem cells. And in fact, they argued that conversion of primed cells into trophectoderm can't take place, but that in essence, the prime cells can, for the most part, only give rise to amnion. So again, at the same time, there were that we were setting out to publish our paper, about 4 other groups had shown that some version of BMP treatment or other media could in fact convert primed pluripotent stem cells into trophoblasts. So we actually set out, and I'm just going to kind of give a quick overview of the data that we showed in this paper, that our cells had a trophoblast gene signature, not amnion, that L5 promoter hypomethylation, which is a characteristic defining feature of early gestation human trophoblast, was present in our cells. And our cells actually did show loss of classical HLA expression once valproic acid was withdrawn from the media. And again, other groups had shown the same thing. The one caveat that I will add, that the one way in which the primed pluripotent stem cell derived TSCs were different from the naive is in their expression of the C19 microRNAs, which, as you all know, is also a very important feature of early gestation human trophoblast. And so we do admit that basically these microRNAs are not as robustly expressed in the primed cells, but we would argue that while these cells are maybe slightly less proliferative than the naive derived cells and primary cells, that they in fact are still bona fide trophoblasts. They meet all the other requirements of the TSC state. So why does this matter? Again, for this, I'm going to quickly turn to describing the work of Dr. Mariko Hori, who's now set out her own independent lab here at UCSD. And this was a collaboration between her lab and Dr. Thor Tunison at WashU, who was one of the people that came up with one mechanism of doing naive conversion of primed pluripotent stem cells. So they set out to basically look at the transcriptome and epigenome profile of cells that in order to model preeclampsia. And the way we at our center here work with have set out to model disease is that we take placentas at delivery, which are very well characterized both clinically and based on placental pathology. We derive umbilical cord mesenchymal stem cells and reprogram these to iPSCs, and then using our protocol, convert these cells to TSCs. Now, what Mariko had already done was that she had derived cells this way from both pregnancies that were affected by severe early onset preeclampsia versus the absence of any pathology. And she took these cells that were in the primed state and before converting them to TSCs, she shared these cells with Thor's group. And in Thor's lab, they converted these to naive iPSCs using their 5-ILA media and then converted them to TSCs. And then Mariko took both her primed iPSC derived TSCs and Thor's naive iPS derived TSCs, again, originating from the same patients. And she looked to see how the resulting trophoblast can model trophoblast differentiation and the disease phenotype. Now, what she had already seen in her primed cells is that the primed cells that were TSCs that were derived from the preeclamptic placentas, they showed decreased expression of HLA-G. They didn't seem to be able to differentiate very well into EBTs. And this would be a hallmark of preeclampsia, as we know that this is one of the early defects in placentation that happens in preeclampsia, in particularly severe early onset preeclampsia. Interestingly, this phenotype was lost when the cells were converted to the naive the naive state and then converted into TSCs.
Mana,
She
can
also,
I ask a question?
yes, of course.
Is it the percent of cells? And did you also notice a difference in the intensity of stain per cell, i.e., was there lower expression of HLA-G, or was it kind of plus minus binary versus a shift?
I believe that this was the actual expression, the percent cells that expressed HLA-G, but I think there was also a decrease as well. I mean, I'm
Okay.
sharing Mariko's data, so I'm
Yeah.
not as
Okay.
familiar with it, but I think it's a bit of both. But what this is showing is the expression, the number of cells that express HLA-G.
Got it. Thank you.
She also showed this, I'm not showing the data here, but she showed an overall defect also in invasion as well. And then this is just to summarize the comparison of the transcriptome. She compared her EVT, both the primed and naive, to Okai's EVT signature. And what she sees is that basically in the controls, in the cases that are not affected by preeclampsia, there's about a 60% overlap between the genes that are in these pluripotent stem cell derived EVT compared to the Okai EVT. But within the EVT that are derived from preeclampsia in the primed state, there is only a 30% overlap, again, confirming this sort of defect in EVT differentiation of these cells. But again, this loss of overlap is abrogated in the naive converted EVT, again, confirming that along with the loss of phenotype following naive conversion, there is this loss of the transcriptome phenotype that goes along with that. So this shows that basically primed pluripotent stem cells can be used to produce bona fide TSC-like cells, and they can retain the epigenetic disease phenotype. We're still kind of looking to see, again, how BMP is initiating trophoblast induction in these cells, as this mechanism is unusual, and we're trying to further evaluate amnion versus trophoblast-specific markers in these cell types. So I'm going to quickly move on to the second part, which is how in the past 4 or 5 years, we've been working to now also differentiate pluripotent stem cells into other cell types at the implantation site. And I'm going to, in particular, talk about our recent work differentiating these cells into decidual natural killer like cells. And this is work that has been done by a talented postdoc in my group, Virginia Chung. So we basically we have a collaborator here, Dr. Dan Kaufman, who has been involved in establishing a protocol for differentiating iPS cells into natural killer cells. And so we adapted this protocol from his, kind of adding to the end of this TGF beta, which I will explain why in a minute. But basically, this protocol is a little bit longer than the trophoblast differentiation protocol. It takes about 2 to 3 months to accomplish this. But basically, by the end of it, we get a uniform population of CD56 positive cells, which during expansion start to express CD16, which is more characteristic of peripheral blood type NK cells. But what we've basically found is that we decided to add TGF beta to the expansion phase and see if this can actually result in a more decidual NK-like phenotype. And the reason we did that is because there are publications showing that peripheral blood NK cells, when they're treated with TGF beta, which is secreted in the implantation site by decidual endometrial stromal cells, as well as other immune cell types within the implantation site, that when TGF-beta is added to peripheral blood NK, they do assume a more decidual NK-like phenotype. So when we did that, we did see that, in fact, expression of CD9 and CD103, which are markers of DNK increase, and particularly CD103, which is almost non-existent in the non-treated cells. Again, this is the percentage of cells expressing this marker significantly increases. And we did a single cell analysis, again, summarizing this very quickly. This is hopefully going to get published soon in Stem Cells, so you can, and it is posted on bioRxiv, so you can see all these data. But basically, the single cell showed that, again, the non-treated cells sort of group over on the right here, TGF-beta treated cells on the left. And when we used the single cell data from Ventotormo et al. To identify what subtypes of NK cells are expressed, basically what we found was that the majority of the cells, actually even the non-treated cells, had a more DNK type phenotype compared to peripheral blood NKs, but that it seems like following TGF-beta treatment, there is more of a shift toward a sort of less activated phenotype of the DNK2 and of the DNK2 compared to DNK1 and DNK3. And so we then moved on to look at the function of these cells. So without any further stimulation, we found that both the non-treated but greater in the TGF-beta treated cells, these cells produce pro-angiogenic molecules, VEGF and PLGF, as expected from such primary cells. And then following stimulation, they do also upregulate CD107A, and they secrete TNF-alpha, GM-CSF, and interferon gamma. So to summarize, we've shown that these HPSCs can be differentiated into different subtypes of DNK. We're currently evaluating their killing function, expecting that they will be functional, but perhaps following TGF-beta treatment, especially in the presence of trophoblast cells, extravillous trophoblast, their killing activity will be somewhat decreased. In future directions, where we have actually banked cells from both from matched maternal and placental cells, and the plan is to differentiate these cells into matched DNK and extravillous trophoblasts to model the implantation site within both matched and mismatched settings. And then very quickly at the end, want to put a plug-in for placental pathology. This has been in particular, I feel like in the past few years, I felt like, I need to go back and make sure that the work that we're doing on the research side is complemented by the clinical work in placental pathology, and that this is kind of plugged in both on the clinical side and on the research side into what we do. So as some of you may know, in 2015, there was an Amsterdam consensus statement that was published on sampling and definitions of placental lesions. That was really, prior to that, there hadn't been very much in terms of standardization of placental pathology. This was further reinforced by a publication in 2021, sort of trying to emphasize how well the Amsterdam consensus criteria could be applied for diagnosing placental pathology, really into these four major categories. I talked about maternal vascular malperfusion earlier, but these are the other sort of three major patterns of injury in the placenta that can be diagnosed post-delivery. Unfortunately, in 2022, some of you may know that this paper was published in the Green Journal basically saying that or seem to say that because many OBGYNs don't understand the nomenclature and placental pathology reports, that, we shouldn't be doing placental pathology, maybe with the exception of stillbirth and possibly unexpected severe neonatal depression. So as you might imagine, this was pretty distressing to the placental pathology community. We did write a response that was published in the Gray Journal shortly after. And more recently, I keep trying to sort of put a plug in how placental pathology can be useful in both the clinical and research setting. The Amsterdam criteria are actually due to be updated later this year, and we're hoping that there will be further consensus-based grading of criteria for these specific patterns of placental injury. I'm also working within the Society for Pediatric Pathology to establish standardized language for placental pathology reports, and hopefully then further educate OBGYNs and neonatologists in the utility of placental pathology. But in the meantime, kind of wanted to put a call out, especially within this group, that we really need to incorporate standardized placental pathology into basic placental biology studies. So I know a lot of people use quote unquote cytotrophoblast preps from normal placentas at delivery, make sure those placentas are in fact normal, but maybe teaming up with a pathologist to take a look at those placentas for you. And we really should be incorporating placental pathology into ongoing cohort-based epidemiologic studies, including studies such as environmental exposures, and particularly in MFMU and NRN clinical and interventional trials that are going on, in order to be able to better use placental pathology to give information to inform decision making, both on in mom and her subsequent pregnancy, but also in taking care of the neonate. This is mostly addressed to patients and patient advocates. Placental pathology is definitely preferable to placental encapsulation, and that it really should be done in every pregnancy complication, and that positive and negative findings can be really useful. And always encourage patients to ask if their local pathologist is using standardized criteria, both for sampling and for diagnosing their placental pathology. And here at our center, which is a focal point for basically all studies that are related to the placenta, we incorporate placental pathology into all of our studies. Most recently, we've been funded by the NIH as one of the stillbirth prevention consortium centers for a project that is titled Maternal and Placental Determinants of Fetal Growth Restriction. And our goal is to identify maternal proteomic biomarkers of fetal growth restriction that are specific to each pattern of placental injury. And this is going to be a CPD-wide collaboration with our colleagues in OBGYN and neonatology, clinical chemistry, and the School of Public Health. And the goal is to have a prospective cohort of patients with suspected growth restriction where we follow them and have detailed clinical placental pathology annotation, proteomic screening of maternal blood, along with measurement of allostatic load through maternal surveys, and using machine learning-based data analysis for risk stratification of patients. And I probably went a little bit over, and hopefully there's still some time for questions. I mainly showed you data today from my postdoc, Dr. Virginia Chung, who's going to be applying for jobs. So if you have any openings, please let me know. Rob Morey, who's our genius computational person here in the group. I showed you work by both Francesca Sonson and Marco Hori, who now have their own independent lab here at UCSD. And a big thank you to all of our collaborators and funding, and I'm happy to take any questions.
All right, Mana, thank you so much. That was great. I think we'll definitely have to have you come back after the updated Amsterdam and have you educate us all on the clinical pathology with some slides and showing us the different scenarios with the new information. That would be actually really, really helpful.
Okay.
So while other people are thinking of questions, um, so it seems to
Joel
me,
has his hand up, Virginia. Just
oh, excellent. Go ahead, Yoel.
Well, thank you. It's fantastic. And Mana, as a non-pathologist, I fully agree with you on the need. We just need to proliferate you more, so we have more placental pathologists. But I guess my question is, there's this discrepancy of information about the significance of the prime versus naive cells. And the Nature Communication paper from Okai said that, he cannot really proliferate the prime cells. He can only proliferate the naive cells. And he attributed this to the C19 MCs, as you mentioned. So 2 questions about. First, I still think that this discrepancy has not been resolved on the significance of the proliferation in naive versus prime cells. And 2, why do you think it is that you can differentiate the prime cells into preeclampsia-like phenotype, but not the naive cells?
I definitely think the phenotype is epigenetic based. And Mariko, if you look at Mariko's paper, she did also do whole genome bisulfite. But I don't know if you've done any of that work. Correlating your whole genome bisulfite to transcriptome is very difficult. They did do some of that and made some conclusions, but I don't think, I mean, there's limitations in terms of what you can do to directly prove that, exactly where the epigenetic phenotype is. But the fact, I mean, I think that this is one reason we got Thor's, interest peaked, that, we could, by doing this, naive conversion and just doing the ultimate experiment of like, can we actually, because the goal at the end of the day, because everyone would keep asking us, well, why do you think why do you think you're right? And I'm like, that doesn't matter. The why actually doesn't matter. The question is, if we're trying to model disease, which one works better? And then you can get at the why. And then, the nice thing right now is that, we're admitting how these cells are defective. They don't, if you look at, and Marco's paper shows this very clearly, primary cells, naive TSCs, and prime TSCs. If you look at proliferation rates, the proliferation rates for the prime cells are lower. And we think, and that should be tested. We've actually, we might be wanting to try knocking out the microRNA cluster and kind of seeing, exactly what happens to proliferation. And that would directly answer that question. But I think beyond that,
I can tell you, Mana, we knocked it out and the cells don't proliferate.
And the cells don't proliferate. There you go. Yeah. But so I see, that's the thing though, is that it's not that these cells don't have any expression, right? They just have a lower expression. And it's, of course, it's also a microRNA cluster, right? So yeah, I think we could look more closely. I think the problem, honestly, is the 2016 What is Trophoblast paper. I have a problem with the way folks are defining trophoblast, because I think that while the C19 microRNAs, as you've shown recently, is very important for trophoblast innate immunity, and it might also be important for trophoblast proliferation, it doesn't necessarily, a certain expression shouldn't have to define trophoblast identity. So I think we have to be careful how we address that, especially in this day and age with the restrictions on, federal funding of, primary trophoblast research. If you want to make people can do naive conversion to study trophoblast, you're not going to have a lot of people doing it.
Oh, great. Thanks. Matteo, you had your hand up earlier.
Yeah, Yoel actually completely stole my question, but it's still related to the prime naive state. And it's very interesting as well that you mentioned about the phenotype. When the iPSCs were, if I understood correctly, from the primed, they show the phenotype of the preeclampsia, while if they were reset back to naive, then they will lose the phenotype.
Yes.
And that's, I think, is also quite powerful in terms of thinking like the intrinsic, the modeling of the pathology, right, and retaining the
Right. Now, I will tell you it's again, and I'm stealing Marco's thunder here. This is all her now. I don't do any of this anymore, but she is right now the question, the why question and the role of the microRNAs become really important. The other thing is, well, if this is kind of going to be continued to not be accepted as a model, another thing that Marco's trying right now is the Jose Polo method of the direct reprogramming, say taking the MSCs, right, and then applying the same, the standard, the OSKM, reprogramming media, but then capturing the intermediate right then in TSC media, and then and that's done in hypoxia. I know Marco will tell you the reprogramming works. It's just a pain in the butt to do all of that reprogramming work under hypoxia, but it is doable to get directly reprogrammed TSCs. And then she's right now doing all the ATAC-seq epigenomic, analysis to basically see if, and also the phenotype analysis to see if that's retained in the directly reprogrammed cells.
I think also it could be, it's super interesting, and I'm so glad also that you clarify the different states and the amnion phenotype and so on, because there was a lot of confusion in the field. So I was so happy to see your.
There still is, and the amnion. I don't think the amnion question has really been, because I will admit there are again some amnion markers that are slightly higher in the primed cells, but the great majority, if you compare using all your data, it's they cluster much more closely with trophoblast than amnion.
I'm glad that you caught up, because many people were making like even blastoids using those cells that in the reality they were like probably amnion spheres or something at that level. Plus, it makes sense embryologically because the prime state that we know the amnion derives from the epiblast cluster. Right. The post-implantation, while the trophoblast lineage comes from an earlier differentiation. But going back to the prime to naive, I think this could be also a very interesting opportunity to potentially looking how the epigenetic remodeling or something that you can do can erase the pathological, can rescue or erase a pathological phenotype, for example, methylation and so on that happens between the naive to prime transition. I think it's very, very powerful, the fact as well that you retain the phenotype when starting from one state versus the other that completely obliterate, in terms as well of understanding the pathology, but also intervention in terms of,
Absolutely. And that's, again, Marco's group is working on this, trying to look to see if, kind of both ways, what if you started with kind of normal iPSCs, but could you treat them in such a way, right? Whether it's with, hypoxia, reoxygenation, or, inflammatory factors, et cetera, to sort of recapitulate that epigenetic and the phenotype and the associated transcriptomic and epigenomic changes.
Thank you.
And actually, Mana, we have one more question from one of our MFM fellows, CC Plummer, who is also actually working hard at creating these dyad, maternal fetal dyad IPS lines.
Hi, Dr. Paraz. This will be really quick because I know we're running over on time. I just wanted to actually ask you if you or Moriko or anyone else have ever tried using other cell types besides the MSCs for conversion into iPSCs, especially from the cord.
So we haven't tried like cord blood stem cells. The cord MSCs have been the easiest to use, but we are starting to think about, the other question that, well, again, Mariko keeps getting asked is why not start with trophoblast? And especially now that we have a protocol where we can take term cytotrophoblasts and culture them for long periods of time in our modified Okai media. Because as you may know, for reprogramming, the cells need to be proliferative. So because we can passage term cytotrophoblasts now in that media, the plan is to maybe reprogram the trophoblast directly. And then this way, that's even more relevant to, if you're trying to sort of model trophoblast disease. So
yeah. Okay. Thank you very much.
Of course.
All right. Well, thank you very much, Mana. Really appreciate it.
Thank you. Sorry, I talked very fast and I also went over and didn't.
You had a lot of people interested in really nuanced questions. So thank you for entertaining them.
I'm excited. Thank you.
All right. I'll see you at SRI.
See you
Bye-bye.
there. Bye.